Good morning, guys, gals. How we doing?
It's Friday, man.
It's Friday.
It's a great day.
Louise Hecker, welcome.
Thank you. Thanks for having me.
I cannot wait to have this conversation. It's like two in a row. We had a great show yesterday that I couldn't wait to have. We have a great show today. I can't wait to have. It really
I don't
is
know,
awesome.
man. Last night's left me with a headache.
Yeah. we talked about quantum synthetic data in biology. And yeah, it's really cool.
And the existence of 37 universes. So
yeah.
It broke me a little bit. It was a lot to kind of keep in your skull, you know?
So yeah, Dr. Louise Hecker, let's talk. Where'd you come from? Like you kind of hit the airwaves pretty prolifically recently with the article that we'll get into. But like prior to that day, what got you to where you're at here?
Yeah. Well, I guess I'm one of those people who came out of the womb kind of being a science person. Always been interested in science from, my early childhood days. Went to college and, started doing research right away as a freshman. I was just completely hooked. Went to grad school, worked, got a few graduate degrees. All my background and training is really kind of focused on, up to my PhD, through my PhD, and kind of all types of regenerative biology. If you look at it as a whole, nothing really connects. It's kind of like everything from
you
like
just described
we love
show.
that go look at the titles of the last 170 episodes we've done.
Yeah.
Nothing connects, but it all connects, so like everything from limb, why are we finding deformed amphibians in the wild? So like limb regeneration and in amphibians.
We're gonna have you back.
Yeah. Talk about mutated newts.
I developed the first bioengineered anal sphincter.
Oh. I mean, that is quite the LinkedIn post.
We can see at least 30 applications for that.
Yeah.
to more toward my PhD studying fibrosis, which is the clinical term for scar tissue.
Mm-hmm.
And that kind of led me into starting to study aging. And I didn't realize it at the time, but having this background in repair and regeneration really kind of cued me up to study aging when repair is less efficient. And so my professional career is really focused on all things aging, why, age-related disease, why don't we repair as well in older age? And my main focus is in scar tissue and fibrosis, which we tend to get a lot of fibrotic diseases in older age. And so understanding why that happens and how can we develop treatments that promote better repair.
Are there a lot of versions of age-related fibrotic conditions?
Oh yeah, so many. So I study a rare disease called idiopathic pulmonary fibrosis, which is the most progressive and deadly fibrotic disorder in your lungs. But even things that you've probably more heard of, like liver cirrhosis, that's scar tissue of the liver. So fibrotic disease affects so many. So I mean, when you die of heart failure, most people don't realize that actually you die of organ failure, but what causes organ failure in the heart in most cases, or many cases, is that you have this progressive scar from where you lost blood flow. That scar continues to grow, remodel your heart responds, it blows up in this area, contracts in this area, and ultimately you die of organ failure. But scar tissue is really the thing that precedes that.
It's weird. We just had a friend of the family get diagnosed. Sad story, but he got diagnosed, and I can't remember the name of it, but it's a fibrotic disease, blood cancer, where the bone marrow turns into like where you're supposed to make cells, you make scar tissue instead. I can't remember the name of it, but it's not good, but it's like a similar thing. He's a younger-ish guy, but now I'm like sitting there going, oh, I'd never heard of this. Like it never even occurred to me. And now, we have you on 2-3 weeks after we found out about this. Mm-hmm. That's wild. So is there like with these things, was your interest more in like codifying them or preventing them or a little bit of both? Like, I mean,
with scar tissue?
Yeah.
Yeah. So my
a bit of a niche rabbit hole now.
Yeah. So what I'm really interested in is how can you develop therapies that that not necessarily reverse, but reduce established fibrosis.
Mm.
So if you have a growing thing, we have therapies now that can slow that progression, but they don't stop it and they don't reduce what's already there. And so where our focus is really, can we reduce that?
Is it a genetic malfunction that causes this, that cascades into something bigger?
It sounds more like a natural process of aging if you have that right damage or something being off.
Well, I mean, everything we manifest is a combination of age, genetics, environment. And so there are genetic risk factors, there are environmental risk factors, and then, aging is, I would argue aging is the biggest risk factor for fibrotic disease.
Seems like aging is the biggest risk factor for a lot of shit. Yeah. You know what I mean? It's just basically like a lot of stuff. Like,
it kind of sucks.
Yeah. 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 So I mean, we've talked about like Sinclair and those guys a lot on the show for the same reason. It seems like everybody's kind of in the last 10 years, certainly in the last 5, I think focus has really gone almost myopically over to like, let's treat aging as a disease and work backwards from that, like as opposed to like, let's treat fibrotic issues or let's treat gray hair. Let's, I
think I like that though. And that makes a lot of sense from what you're saying, right? If aging leads to more fibrotic issues, it's going to lead to brain development stuff, cognitive function, clearly a whole slew of physical stuff. And to be able to undo that or extend your lifespan makes perfect sense that you're going to delay that fibrotic process. I know that's probably not the right term, but
yeah, no, it's right. I mean, can you please go tell the government that? Because
I got some other things I want to say to the government.
Yeah, I was just going to say, Louise, I've got a whole bunch of shit I'd like to say to them.
Believe me, if I move my head to the left or the right too much, you're going to see some hammers and sickles.
Yeah.
I'm done.
I couldn't agree more. I mean, I think that studying these processes is fundamental to understanding, in the context of age-related disease, that's what we need. One of the tough things as a scientist is it's a lot tougher to get funding to just study aging. It's a lot easier to say, I'm going to study this age-related disease.
Australia,
is that kind of how you got your toe in the door, your foot in the door there?
With the Psilocybin project?
Yeah.
Well, it really started with just a friend of mine who just is crazy about psilocybin. He's always asking me questions about psilocybin, and my answer is usually, I don't know. How does it work? How does this work? Does it do that? What's it being used for? And after being asked questions for months and months and months, I finally was like, all right, I will read about it and report back. And so that's actually how this whole thing started.
Fine, I'll take a bunch of mushrooms.
So I'm going to start hammering you on LinkedIn with questions.
Yeah. And this is how exciting ideas start, right? Like someone just asks you a question, and as a scientist, I wanted to be able to answer scientifically. I didn't want to, because he's getting his information from, no offense, but podcasts.
We claim no medical knowledge whatsoever. Please do not worry about that.
As scientists, we can only get information from peer-reviewed articles, and that's the only information that I'm allowed to use as a scientist. And so I wanted to find out from the literature what's really known, because I didn't know, and so I started reading and just reporting back about what I was finding, and honestly, once I started reading, I mean, I was just hooked right away. I was just so fascinated by what was out there, just this mushroom that is being used for so many diseases.
Did you have any experience with psilocybin and be prior to that?
Absolutely nothing. I knew nothing about it, but kind of the basics of what everyone knows from social media and Netflix.
That one crazy day in med school.
Yeah, like that one Fish concert I went to when I was 19.
Yeah, exactly.
Yeah, that I knew the basics, but that's about it. So it was interesting to start reading about it scientifically, and just as a scientist, my interest was piqued. Like,
sure.
Wow, there's so many diseases it's being used for.
Mm.
One of the things that was intriguing in particular was, people were reporting these long-term beneficial effects. I mean, from one dose, 5 years later, they're having positive impacts. So from a scientific standpoint, that's mind-blowing.
We don't have too many drugs that do that.
We don't have any.
Do we have any drugs that do that?
Yeah. Any drugs that do that? I'm curious. I can't think of any.
We talked to I met the guys from the psilocybin lab at Hopkins a few years ago at an event, and we were just kind of bullshitting after the event. And he was so cavalier about their results, probably because he's doing it like day in, day out. It's no longer, he's like anesthetized to it a little bit, but he's like, yeah, we found out that like 90% of people that come here or something, I'm making up the numbers, like quit smoking. So we just started another study for smoking cessation. And he's like, it just keeps rolling off his tongue like the next weird thing that they did.
But isn't that what like great science is, right? It's the curiosity. It's like, well, that was weird. What does this mean?
Yeah.
Speaking of which, we got to get to a PhD in pints, sponsored by Hopkins here. I meant to bring that up to you completely aside.
Yeah. Okay. Sure.
Sorry about that.
It is. I find this to be very fascinating in general because I think it's because it works and we don't really know why it's working. And I think work that you're doing and stuff is great to kind of nibble away at that. But like, why do you think we don't know so much about this? I think it's pretty clear we don't know way more than we know. You know what I mean?
So true. That's so true. And so, one of the things that we've done recently, we haven't published this yet, but we're about to put this out. But we did what's called a systematic review, which is like where you ask a science librarian a question and you answer it. And the question we asked is, who has done psilocybin research and who has funded it? And what we're finding is interesting in that there are very few articles. I mean, there's like 300 something articles total ever published on psilocybin research in the United States. And a very, very small fraction of those studies were government funded.
Wow.
So it's been tough. Now, why?
So private sector, is that who's funding most of them?
Well, do you think that's because it's still classified the way it is nationally?
There's so many reasons that play into that.
Okay.
So one being that it's a Schedule I drug.
Sure.
That's a big one. That's a big one because because when it's scheduled, so it's the same as heroin. It's the same class as heroin,
which is nonsensical on every level.
Right. So when you say, so when you go into the lab and you say, I want to study a Schedule I drug, well, number one, there's a huge process. You can't just walk in the lab and the next day you've got psilocybin in your lab. You have to go through all of these things. You have to get a DEA license. You have all these regulatory barriers. You have to have the psilocybin in a lockbox, inside of a lockbox, behind a lockbox.
That's crazy.
I got a white dude with dreadlocks that can get it for you.
Yeah. There's a guy who hangs out at the Stop and Shop. He's got
And the fact that it's even legalized in some places, it's just puzzling, right? So there are incredibly high regulatory barriers that prevent people from, I don't want to say prevent, but make it extremely challenging to do psilocybin research.
Sure.
And I'm assuming that raises the cost exponentially.
That raises the cost, right? So you have to go through all this entire process. So even if like in the lab, if we use psilocybin on cells and then the cells touch the psilocybin and we take whatever touched those cells, we have to send to a company to reverse destroy it. So we can't. So there's so much of a regulatory process so can you imagine as a scientist?
So after the show, Tom and I will give you our addresses. You could
where's that dumpster?
Don't you worry.
I'll give you something certified.
Yeah. That is insanity. Like what an epic waste of money.
God, and how anti-intellectual.
Yeah.
Well,
in general.
I mean, I agree that it's, so the classification as a Schedule I drug certainly make it really challenging from a scientific perspective. The other thing is, when you, at least in my experience, applying for grant funding. I mean, you apply for grant funding and then, I just got blown out of the water initially. I mean, people were like, one of the critiques is like, yeah, it's a Schedule 1 drug. Well, that's
that bluntly, huh?
no kidding. I mean, so that's not the point, but that's being used against me in my, so it's really difficult to get something. It's been difficult to get funding for this. And if you look at the percentage of funding that the government gives to psilocybin, it's very low compared to a traditional drug that we're developing.
Is it
for profit?
Does it make it easier or harder or no difference whatsoever? Like Texas just basically legalized Ibogaine, for example, right? So if you got hot to trot on Ibogaine, like, is it easy just to pack up stakes and move to Texas for a little bit, or is
I actually am in Texas now.
Oh, wow.
Oh, you are. Oh, perfect.
I'm hearing about this a lot, actually, and yeah, I think. I mean, I'm going to study. I'm now interested in all these.
Sure.
But yeah, I think as these things happen, it will make it easier. I mean, I think it's just a matter of time. Well, I guarantee that it will be reclassified soon. So the main things that make something a Schedule 1 drug is that it has no
Therapeutic value,
medical use,
Right. Which is changing all over the place.
Which is changing. I mean, it's already in phase 3 clinical trials for different indications. So if it does get FDA approved for any indication, then it suddenly has an accepted medical use. I mean, the other thing is a high potential for abuse. I mean, it is actually being used in clinical trials.
For addiction,
For addiction, yeah, exactly.
So that one's puzzling to me. And then lack of an accepted safety profile. But the FDA has given psilocybin breakthrough therapy designation,
Sure.
which is something that underscores the safety profile.
Correct.
So all of these things don't really make sense anymore. So it's just going to be a matter of time before it gets reclassified to a new schedule. I'm excited for whenever that happens. That will lower this regulatory boundary kind of threshold of the challenges that people like me face for studying it. And that will open up a lot more doors, I think, for people to come in and more readily and easily conduct this kind of research.
Yeah, and it's starting. I just read an article this morning that Arizona's doing an Ibogaine thing too, and so it feels like once one domino falls, that like a bunch of other ones start falling right away. Like they were just waiting for somebody to give them permission or something. It's like, all right,
it's
Texas
so crazy
we're good.
too, right? I mean, cannabis is still not legal in certain places, and now it's like, no. Let's go to the ones that will make you have an ego death. Like it's like there's no, we don't Americans, I don't think, have like a middle of the road view on anything.
Yeah. I just saw Czech Republic is actually looking at Ibogaine and esketamine. So it's like a weird, like places are popping up all over the place that are kind of looking at these substances. And I'm like, there is a part of me that's like, oh, well, AI's here to take everyone's sort of jobs. And that's great because we'll all be on psychedelics.
We'll all be tripping.
so it'll be all cool, man. We don't have to worry about it.
Yeah.
So how does that lead to, did you stumble on the aging thing by accident kind of thing? Like, how did that end up popping up?
Yeah. So I'm reading all this stuff. I'm reading about all these different disease indications that it worked for. I'm reading that it's has durable effects, meaning you take one dose and it lasts. And I'm just getting more and more curious. And I stumbled across this paper that had an idea, a theory, if you will, that, maybe if it's doing something to aging, and if it's impacting aging processes, then that's a mechanism by which could help potentially explain how it's having these durable effects, how it's working for so many disease indications. And I just thought,
and what's the hypothesis there? If you have a durable effect, you might have rewound the clock a little bit on some of these sort of like age-related things. Is that kind of the thought process?
Yeah, like if this is something that impacts aging processes at the molecular level, then that's something that can have, I think, can contribute to more long-lasting effects than just if you can reduce hallmarks of aging, or then like for example, if the idea that I read, which again there was no evidence, but somebody said, well, maybe it impacts telomere length.
Mm-hmm.
And telomeres are, you know, the protective caps on your DNA, and they're important because they provide protection of your DNA, and so if you have more protection of your DNA, then that's going to lead to all kinds of downstream impacts. And so I realized, as someone who studies aging, that this is my wheelhouse. I can test anything age related, so I can actually go in the lab and test that, and that's what I set out to do.
That's awesome. And like, were you guys, I mean, it looked like you went in with a primed pump a little bit. Like, I mean,
the hypothesis was pretty solid.
Yeah, the hypothesis was pretty solid. Were you guys kind of surprised at what you found? Like, were you kind of like, shit, we should test this a few more times?
I wouldn't say the hypothesis was solid in that. Like, the hypothesis, it's like an idea is a dime a dozen. I mean, you know, it was somebody's idea. That said, I was like, this is a crazy idea that's really intriguing. And if it's true, then that'd be really exciting. And so I went into it just with like, you know, well, I'll see what the data says. And and so we have, you know, ways to study aging in cells. I was able to get pilot funding from Emory. I started these studies at Emory. Emory was very generous with giving me they gave me one round of pilot funding to do the cell work. And then I was able to get another round of pilot funding to do the animal work based on the exciting cell data. So the cell data came back, and I was just like, I mean,
Get into a little of the results here. Yeah, tell us a little what you observed.
Yeah, so when you study aging, one way that we do that in cells is these models of basically cellular aging, which you take cells and you just literally age them over time, and you can follow these cells over time and quantify how old they are getting, essentially until they become old and senescent. And as this process is happening, you can follow that. You can see all of the age-related changes. So it's a very well-established, validated model to study cellular aging. So the experiment that we did was very simple. Just let's age cells with a vehicle, and the other group had we use psilocin, which is the active metabolite of psilocybin. So when you ingest psilocybin, it's broken down, and the active metabolite, which is what the rest of your body sees, is psilocin. So we gave cells psilocin, and we saw what, you know, the major endpoint was what happens to cellular lifespan. And so we found that when normal cells kind of growing and reaching their peak and aging and senescent, these psilocin treated cells were still dividing. They were dividing faster. They divided more. They were living longer. And then they did get to their senescent, you know, point, but it just took them much longer. So this seemed like the cellular aging process was significantly delayed with the cellular treatment.
So I'm not sure you went there, but was this something that like additional dosing had no additional effect? Like
No,
there was a cap?
We did do that, actually. We tested this in a dose-dependent manner. So we had like, you know, a low dose, a medium dose, and a high dose. And the more psilocin you gave, the more impact on life extension.
Oh, shit.
So we gave 10 micrograms.
That is calling his plug.
Yeah. Well, wow. Yeah. Well, no, I mean, I'm interested because, and again, I can't believe that when I watched this movie 100 years ago that I'd quote it so much on a podcast one day, but there was that movie, Venus Rising, I think it is, or Jupiter Rising. And like, they took this like thing and then they jumped in a hot tub of it every so often, and it kind of restarted the clock, right? And it's like kind of one of those things, like, is this something that I could take one on a Monday and then a year later I could take another one and I roll back the clock to the same thing? Or is it basically like, yeah, you get that initial kick, but that's it, like it's like a one and done situation?
Yeah, I mean, all these questions we need to figure out.
Gotcha. So we're still.
And are we still in a cell model? We're not even on like rats or human trials?
Yeah, so we
Yep.
did this, and so once we saw this in cells, and then the next thing we did once we saw the major finding, which was okay, cellular lifespan is extended. We started looking at what are the molecular mechanisms? What is it impacting in the cells? And that's where we saw well, telomere length is preserved. We saw DNA repair markers of DNA repair were improved, or evidence that there is better DNA repair.
Wow.
We saw oxidative stress levels were like lower.
So multiple methods of action from this.
Multiple markers of action. We saw less senescence. We saw all of these things that were intriguing in the cells, and that's what gave us that's when we said, well, we have to take this to animals now. And so we did the animal study, and the animal study, we wanted to design something that was really the most realistic, clinically relevant protocol. And so what I mean by that is we wanted to think about if you're one of us, what kind of evidence would you want to see? And so we thought, okay, let's start with older mice. Let's start with mice that are the equivalent of like a 60 to 65-year-old person, and let's just start dosing them. And so we gave them 10 doses total over once a month. So the mice were 18 months old or 19 months old, which is, again, that in mouse years, that's like a 60-year-old.
Yeah.
And so once a month, I don't know exactly what that translates to in human years, but once a month for 10 months. Now, again, talking about what you asked earlier about, like, why is this difficult to study? And so I wanted to study, let's study lifespan extension. Does it extend lifespan? But the IACUC, which is the body that governs our animal care, they wouldn't allow me to do that study. So
what was the issue or pushback?
Yeah.
So it's again, I like using the example of heroin. It's like saying, because it's easier for people to conceptualize, it's like saying, okay, I'm going to give super high dose heroin to mice for the rest of their lives, and there's no evidence to support that this could work. And so they're like, that is just so they thought that's not really a good starting point study. And, and I can understand this is a risky study. There's zero evidence aside from our unpublished studies. So they work with you to design a more appropriate starting point to test, because their goal also is the welfare and protection of the animals. And so without any evidence that this could work, that seemed like a long stretch for a study. So what they allowed me to do is to test until median survival. So mice, like people, they have an average, they only live certain, we have average lifespan in mice. And so they said, take this, you can treat the mice, but when any given group reaches 50% mortality, you should have your answer about what it's doing to aging and survival.
I see.
So we did the study until, and it took 10 months for the vehicle group reached 50% mortality, which is just mice that will die with normal aging. They were 28 months old. And at that point, when 50% of the control mice had died, only 20, let's see, was it 20% or only, can't remember. I should know the answer to this. We had only 30% mortality in the psilocybin treated mice. So there was this very dramatic improvement.
So quantifiable difference, like not a casual.
Significant. Yeah.
It was a very significant finding, and that you could see, and so that now, of course, that suggests that this could impact, life extension or lifespan, but we don't know the answer to that just yet.
So did this pave the way for a round 3? Like, are you guys going into human trials or more mice trials?
Yeah.
Like,
oh gosh, well, there's so much to do next. I mean, I think again, like, so as you kind of mentioned, I mean, aging is not a disease, so I don't know how you can do, like, I don't know if it's possible to do like a clinical trial for aging. Per se.
You could, but somebody else has to take over the research at some point. You're talking cohorts, you're not talking a short lifespan on a human,
Right.
But I'm wondering though, I mean, I think part of this problem, if I'm probably grotesquely reducing this, but I think part of the problem is exactly that, Tom, is like that aging is a long process.
Right.
But if we have an established cellular aging sort of litmus test, could you just sort of test their cells on a micro time period and extrapolate over time, just knowing how that's happening? Or does that not work like that?
No. Well, I mean, people are people, and we have, while you can get a lot of information from cells, I mean, people have metabolism, and there's, I think that one of the things I think it will be important moving forward is to evaluate potential harms. And, because at least in the clinical trials that I've seen or that I know about, they've given high dose psilocybin, but usually it's like 1 or 2, maybe 3 treatments. I don't know of a study off the top of my head, at least, where they've given high dose psilocybin long term, repeatedly.
Can I ask the equivalent? Because you said once a month for 10 months.
Mm-hmm.
What is that dose equivalent for say an average human, right? Because
Yeah,
you're saying
I'd have to crunch the numbers. I don't know off the top of my head, but like maybe roughly like once a year.
Okay.
Right.
But yeah, but I think, but again, this is something that really has to be kind of rigorously tested, I think, at the preclinical stage to determine what is the optimal treatment protocol. Like, what if you just gave it to mice once? Would you still see the effect? What if you gave a lower dose? What if you gave it once?
Yeah, lower dose more frequently, or
right. What if you gave it like the equivalent of once every 5 years for human? I mean,
yeah.
What's the dose that leads? What's the lowest dose that you can give that leads to maximal efficacy? And then along with that, what are the things that we need to monitor for potential harms? Is there an increased risk for cancer, or we just don't know, because there have never been studies to really look at that.
And even things like what would happen if you started in younger mice.
Sure.
That seems super cool to me. Could you prevent some things or stave off that for much later?
Absolutely. I mean, questions that I am so eager to test.
Yeah, for sure. Yeah, I mean, it's funny. Sinclair was talking about that all the way back in his book Lifespan about the fact that just because the single fact that it is not aging is not classified as a disease prevents so much progress.
Absolutely.
Like, it's just one little thing. Like, and what is the pushback globally? Because this seems to be an every country problem, not just here. Why aren't governments, if this is a roadblock, why aren't they unroadblocking it? What's the downside of this?
I wish I could understand like how the government makes these decisions and defines these. I really wish I mean, like I said, if you want, I mean, I agree with you. I agree with that statement completely. I mean, I do think that it should have more weight to study aging, because to me, as someone who studies aging is the underlying risk
cause
factor
of all these diseases.
Yeah, totally.
Yeah. I mean, just think about COVID. Everyone, COVID impacts all ages, but without question, you have way worse outcomes with increasing age.
Yeah.
So why is that? And understanding what is different and unique about what happens with aging is important, so that you can understand how to target those things, or helping to prevent, or delay or slow the aging process may also improve outcomes for that and many other age-related diseases, but it's really not prioritized in that way from a government standpoint.
And I think COVID's a great example of a lack, like a realization of how much you don't know about a topic. With COVID, it was the thing I found most fascinating is some people are dead in days and other people get it and they never even knew they had it. Right.
Right.
And I'm like, that's gotta be something. There's an explanation for that somewhere. I don't know where it is.
No one's going to fund that, even though it took out 1 million Americans.
Right.
Yeah. And not even mentioning the long haulers.
And
the significant
like,
problems that people have possibly for the rest of their lives.
Right. And psilocybin research is interesting because it is so like immediate on the therapeutic side, right? Like I have not heard, I'm certainly not encyclopedic, but I mean, we do talk to a lot of people in this space. I've not heard of any story where people are like, yeah, I did it, didn't do anything. Like I've never heard that, like not one time.
And
right?
it's really surprising when you think of the vast amount of study and research being done, and like the social media hype, the regular media hype around psychedelics and kind of this psychedelic renaissance. And you're saying, oh, there's only 300 studies. You know, that seems like what?
Well, here's the other side of it, though. Outside of like Hopkins and stuff that Louise,
you need somebody with the fortitude to be able to actually fill out these forms, jump through the red tape, get the study, funded and approved. It sounds like those barriers to entry, like God, we could do almost anything else, but this seems to be the taboo.
Doing the animal studies, I mean, this went all the way up to like the ethics committee.
Sure.
And, people are weighing in on what I'm allowed to do. So, I mean, it was really challenging from a research standpoint.
So was this study, what you're saying is like you could go on C-SPAN or CNN and watch them debate the funding of this bill? Is that what you're saying at the ethics committee?
No.
Okay.
I mean, like,
okay.
No.
Okay.
I mean, like ethics committees at the universities. Like,
okay. All right, good. Because like, if you let the government do it,
yeah.
The intelligent people work for the government. They don't run the government. You know what I'm saying?
Right.
Yeah.
Yeah. But I think the schedule designation is a huge, I mean, that's got to change, when that changes, that'll change a lot because then there would be more access to it. But yeah, the government does have to prioritize funding. I mean, they have to decide this is important because as you know, I mean, they allocate money towards certain things, and then that money has to be used for these priorities that the government defines. So if the government does not define psilocybin research as a priority, they will not give out the funding. And if if people like me can't get funding to do the work, then how are we going to do the work?
So I actually that tees up my one of my next questions like perfectly is, you look at Hopkins and like what's his name gave like millions of dollars to it.
Tim Ferriss.
Tim Ferriss, yeah. That wasn't government based at all. Now Hopkins obviously I'm sure has.
Well, I think they can score some big name donors.
Yeah, but I guess that's my point. I mean, are people in your field? Because like, look, research, and I'll put that in giant air quotes here, in the psilocybin space and in the world that Tom and I play in is, you got people that say, I'm hurt, I'm sick, I'm fucked up, something's wrong. I know that there's thousands of year olds,
the traditional part of it.
I'm going to outsource my research component to saying, hey, they've been doing this for literally thousands of years. I'm going to trust that this works. And then some dude charges them 20 grand to go on a weekend retreat to, Central America. And then they go do a bunch of ayahuasca and come back and they feel great. Right. But like, that's not research. That's like,
that's anecdotal at best. Yeah.
At best. Right. But like, that is where I think 99% of the quote unquote research is happening is people coming back in droves, like oceans of humans coming back being like, I'm fucking amazing. But like, so why is it like, what's the blockade for people like you to just kind of open up like strictly private sector avenues to do this outside of an academic environment?
Well, the thing is, we can do this. Anyone, like if anyone
you can apply for a study funding,
to say, hey, Louise, I want to fund you to do X, Y, and Z, they are welcome to. I think the challenge is until like now that I have the paper out for me, that I call publications are like your street cred. You know?
Totally.
Yeah.
Because it's peer reviewed. I mean, this is, this has gone through a peer reviewed process, and other scientists say, okay, we can now publish this. So now that I have some street cred on this topic, but up until then, I mean, keep in mind, like at least in terms of studying aging, I mean, there's nothing out there. There's no studies ever, no experimental evidence ever So, and so,
Fucking
from that
mind-blowing.
standpoint, I can
Yeah.
say, oh, I have this idea. No one cares. I mean, I have a million ideas. There are a lot of ideas out there. So I think now that there's a published study, I think a lot of interest, a lot of people.
Right.
Well, I mean, you clearly see it on a professional platform like LinkedIn. I mean, God, even before, I asked you, I think I had probably seen 5 or 6 posts about it from different sources.
Yeah,
And you're right. The idea of publish or perish is really
like posts and Instagram posts, and so it's definitely it's interesting how it's gone so viral.
Yeah, you're like internet famous now over this thing. So it's not just peer reviewed street cred, but you're like actually got people talking about it, which honestly is like I got to imagine in the court of public opinion with the topic like this, it's probably at least equally as important as the peer reviewed side, but if not, maybe a little bit more.
I mean, especially on the funding end, right? If you're going out to pitch somebody, and let's say somebody on that board that's giving out those funds then says, "Oh, she's all over my Instagram feed,"
right?
Like you're like, "Oh, we know you. You're a hot item. Let's invest in this."
Well, and that's the thing, the dudes with all this money who might fund this privately are probably not reading JAMA or AMA or Nature or anything like that. They're probably reading Instagram and Facebook. And until you pop up on those things, it's not going to even get to there.
I saw a great summation in an Instagram post, and I'm like, that's not a psilocybin mushroom. Like the image they use, that upset me.
Yeah.
That's not psilocybin.
I've seen fake news about this already.
Yeah,
that's
great.
That's awesome.
Said to me, "Wow, you really made it when you are fake news now."
Yeah, totally.
So it's gone to viral to a level I can't even believe, to be honest.
Yeah, no. Because look, I mean, I think what makes this fascinating is not, I mean, look, I'm a science nerd. I could listen to facts and figures about cells and senescence and all this stuff literally until my ears bleed. I don't care. But I think there's also sort of like a cultural curiosity when it comes to this, right?
Yeah.
Like it is something that we as a species have sort of disconnected from in most cases that we had previously been connected to for thousands of years. Like this is not like new information, right? And I think that's like kind of intriguing to people, right? And I think it's cool that we're sort of back at it, but I mean, I
think I said that last night, didn't I? Like how the fuck did they know this, right? They probably didn't have the facts and figures that if I eat this mushroom, it might make me live longer. And I understand that's not what your research is saying directly. But at some point, somebody said, here's this thing growing out of this pile of cow crap.
Right.
Let's put this in our mouth.
Yeah.
How weird is that?
Yeah.
And now, thousands of years later, we're having a conversation about, that might actually make you live longer.
Yeah. Well, the other thing is like we when we think of psilocybin mushrooms, I mean, the first thing that comes to mind is the psychedelic impact. And so there's a lot more known about what it does to your brain, mental health, things like that. But we're just scratching the surface on what else it does. And I don't know, no one has really, there are just a handful of studies that really studied what it does.
Other systems.
Yeah.
The rest of your body. There's nothing on aging. So I think because of the psychedelic impact, the focus has been kind of over here. And I think one exciting thing about what our research will do is to open people's eyes that there are so many other things it can do potentially that we just have not studied yet and haven't uncovered.
Yeah.
So crossover question, you guys worked with psilocybin. Was there a particular reason, like is there a mechanism of action inside of psilocybin that would lead you to kind of start there? Because I'm curious, could this happen with LSD, MDMA, ketamine, iboga, or ibogaine?
Yeah, good question. The short answer is I don't know. I think, one of the things, so we know that that psilocybin acts on the serotonin receptor. That's one thing we know. But one question I have is, and the other thing I'll point out too is that serotonin receptors we think of the brain, but serotonin receptors are all over
the body,
the rest of your body. So it makes sense. but the other thing we don't know is, is it working through the serotonin receptor to act on these aging, impacts that we're seeing? Does it act on other receptors, and does it do other things to cells that we haven't tested yet? So we're trying to work on, how is it working? And when we understand how it's working, then we can understand that in the context of, other drugs that are out there and how they might work as well.
Yeah.
Yeah.
Is that the next step for you? Is it like, kind of where are you personally going with research?
Yeah. So we have, so from this, we decided to just get the finding out because we were so excited about the actual finding. And, it wasn't just the survival and the, showing that there's acting on these hallmarks of aging. But if you look in the paper and you look at the pictures of the mice, the mice look better. They grew back, their white hair that was white grew back brown, and bald spots grew back in hair. And
well, one of us has no hair and the other one's almost completely gray.
I'm not a rat.
Keep talking. Yeah.
And there were a lot of things that we couldn't even quantify because, again, I was kind of doing the things that I know at the time, but I didn't know how at the time to quantify necessarily their health span. So I think, that there's a lot to be, I think there's evidence from what we've shown so far that this may improve not just potentially longevity or survival, but health span.
Right. Which is honestly the only thing I think most of the people we talk to are concerned about.
Well, and then you got to look at systems in that. What is it? Cardiovascular health? Is it kidney function? Is it liver function? Digestion, metabolism, all of those things.
Right. So there are a lot of, the study opened up a lot of questions. So we decided to get the finding out. That said, we collected so much more data that we haven't just yet published because it's a lot, and we're still kind of working through what do all of these results mean. So we've taken an omics approach. We have data on from this year-long study epigenetic mechanisms.
Oh, sweet.
We did bulk seq, so we have all the organs and all the genetic profiling of how the genes are changed in every organ. We have proteomic profiling, metabolomics. So we have all this big data.
Wow.
Now, I'm not a person that's not my, bag of tricks. So I have to get other people to come in.
To then interpret the data.
The data part is probably crazy.
But this one study is going to lead to a lot of other papers is what you're hinting at.
Yeah. So we have kind of a mountain of data behind this initial finding that we're still processing. We also did, I'll give you a sneak peek of something else that we did, a microdose group too. So we're still processing the data from that. So that'll be really interesting.
I love that idea.
So part of what we're doing now is continuing to take these samples. We also have, I'll mention, we've done proteomic profiling from a 10-patient clinical trial. So we have human samples, and we're trying to collect a better understanding of what's happening in humans, at least in terms of how it's impacting aging markers. So we have a lot of stuff from that study and the pilot funding we were given at Emory that we're still processing. And a lot of this is also kind of going out, and I love that the excitement and the kind of press from the paper, because what it's really helped for me is to bring in a lot of people who have expertise like epigenetics background and other expertise that aren't, again, my bag of tricks, are reaching out to me, and I'm saying, hey, I have all of this cool stuff. Can you help me analyze it?
Right.
So a big part of what we're doing now is continuing the analysis on all the data we collected from that initial finding that we published, and then designing bigger, huge studies that range from collecting samples in longer long-term samples from clinical trials that are ongoing for other things, to how do we identify the best dosing regimen and protocol that could translate to humans, to understanding the mechanism, to looking at if you take away the psychedelic impacts, can you still have these impacts on aging? So we're doing all of these different, we're kind of tackling it from so many different angles certainly trying to get funding. So again, anyone is welcome. So even though the government does
to write you a check,
anyone is welcome to write a check, and we will research any aspect of this that you're interested in.
I was actually going to ask that earlier, and I forgot about it because we got onto something else. But like with the dosing that you guys had, I'm sure it's just throwing darts at a dartboard at this point. But like, was it psychoactive? Like, in other words, were these mice tripping balls? Like, I mean, is that like, or had you been able to isolate that out? And I guess the follow-up question is, we've all heard the saccharin studies from like the 80s where like it gave rats cancer, but in the human equivalent dose was like 3 kilos or something ridiculous. And you're like, all right, well, I'll be sure not to down a bucket of saccharin. So like, I mean, is there sort of any of those two things you could talk about real quick?
Yeah. So, okay. Dosing is actually one thing we spent a lot of time thinking about when we were putting this study together, because, again, we wanted to try to come up with something realistic. So now it's tough to think about this when you're thinking about mice and humans, of course. So we drew from what we knew from clinical trials. We wanted to give a high dose. We wanted to give a psychedelic dose. So we looked at, okay, what are the doses being given in clinical trials that are psychedelic dose? So that was kind of like the starting point. Okay. But then there were a lot of other factors that play into it. That and again, if you take the actual dose in the paper and try to translate it, I think, and I've seen this online, people are like, that's like 10 times higher than the highest clinical dose. And that's true, it is. But what people may not understand or appreciate is that the mouse metabolism is like way faster than the human.
And was this orally, or was this sub-Q? Like how, because clearly the first pass and everything is an issue.
We gave it orally because, again, that we wanted to kind of model how it's being given clinically now. So we gave it orally.
And was it full mushroom, or was this also?
No, this is like pure medical grade psilocybin.
Yeah.
So
the same stuff Hopkins is using, I think.
Pure medical grade psilocybin, and then you just put it into a I think we use saline, dissolve it, and then we give it to the mice via oral gavage, which is basically kind of like a feeding tube, and you just squirt it in. And we know, so we also drew from prior mouse studies as well. We know a lot about the pharmacokinetics, meaning, the half-life of the drug in mice and humans. We know from other studies in mice what doses have been used to give a psychedelic dose to mice. So we know the mice had a psychedelic dose, not only because we use a dose that studies had previously tested and shown that was a psychedelic dose, but one of the ways that you can tell if mice are experiencing hallucinations is they have what's called a head twitch response. And so we had like GoPro cameras over the cages to monitor. So we can actually quantify that they had this head twitch response. And then actually the mice I was really excited the day we first started this, like, what are they going to do? And they have a little bit of this head twitch response, and then they just go to sleep.
Oh, really? So they're not requesting jam band music and stuff?
They're just getting cozy in the cage and falling asleep after that. So yeah, they did have a psychedelic dose. But anyway, in terms of how do we choose that dose, we did try to factor in the fact that, again, if you give it to humans, it's eliminated a certain rate. That rate is about 4 times faster in mice. So we wanted to.
So you got to multiply the dose.
Yeah. So we wanted to account for, the much more rapid metabolism that mice have. So a lot of factors, including body weight and things like that. So, so it's tough to make an exact.
Sure. I was just curious if it was like, at this point, you're just trying to figure it out. So you just like throw an ocean on them. It's fine.
We definitely knew, like, we want to give a psychedelic dose, and we wanted to give something in the range of what would be equivalent to what's given in clinical trials now.
That's so interesting, because years ago I watched two acupuncturists argue over a study about caffeine prior to acupuncture, because caffeine somehow disrupts this adenosine process, right? ADP to ATP. And I'm looking at the study and I'm like, they gave a mouse a 75. That's like making a mouse drink an entire thing of espresso. That's me getting a 5-gallon bucket of coffee. Yeah, and I was going, that's insanity. Why would we even think that's good? And now that you're telling me it's 4 times,
right, so
like they metabolize 4 times faster, that may be the reason that they did that.
Yeah, and also I think, the other thing to keep in mind is that when you're studying things like at the preclinical level in mice, we're really trying to find an effect. And when we had limited pilot money, because the government wouldn't fund this project at the time. So we had one shot, and
go hard or go home.
So yeah, you sometimes for these initial studies, you're like, I'm looking to see if there's an effect. And so that doesn't mean this is the dose that should be translated. It means this is a starting point to see cause and effect. And then that's where more research is needed, right? Because we need to define the therapeutic index of, how well it's working in the context of its safety. So that all needs to really be rigorously tested and evaluated before we can say, all right, just go take it in humans. So for us, we wanted to see an effect. We want to give a psychedelic dose. We wanted to see an effect. That said, I think it's going to be important to find what is the minimum dose you can give to get similar effect.
Sure. Yeah, humans don't go through that level of effort to determine their own dose. That's for sure. People are just like, yeah, sure, this looks like okay. What do you
this
take?
Seems like enough.
Yeah, this seems like enough. Which which is funny because we laugh about that, and certainly my indoctrination to psychedelics was in a collegiate environment, had nothing to do with no one.
Collegiate environment, not a collegiate study.
Yeah, not a study, no formal study here. And so it's easy to claim Grateful Dead or Fish on this one, and do it. But I love that we're actually asking these questions because when you're doing that stuff, you don't really think, oh, look, my telomere length is going up, or whatever, or, you're not asking questions like that. It's really all about that serotonin response. And you're like, ooh, sweet, and so I'm the nerd in me just shines bright on these conversations because I'm like, dude, there's people asking real questions on this. That's crazy. That's amazing. Mm-hmm. And getting real answers, which is actually even better.
And then of course those answers, make more questions, which is super cool because I could sit here and go, oh, I want to know this now I want to know that I want to know that.
I also think it's somewhat weirdly validating for everybody who is an enthusiast for psychedelics to find out that it's not just good, but it could be really good. It's just sort of not a casual oh, that was okay. No.
It's true. And it's like, I also hear all these anecdotal, all the anecdotal evidence, and I'll send you a link actually to a PBS special that they did on a patient that went through a clinical trial at Emory, and it's an incredible story. And it's just you could see in the patient's eyes that he just looks
different.
He just looks better. And so it's an intriguing drug that deserves more research, I think. And so I if anything, I hope that this opens up more doors for more research to be pursued.
And it's been 58 minutes and we haven't said AI yet, so I'm just gonna
might as well do our due diligence, right?
But I'm curious here. Is there sort of an industry popping up in your sphere of companies that are focusing on AI models to help accelerate your data? Because it's data aggregation has been a problem in research as long as I've heard how people do research.
Yeah.
Crunching data sucks. It's just not, it doesn't seem like a fun process.
Matt had to hit the holy trinity of our podcast.
Right. Yeah. Psychedelics, AI. Yeah. But so how does that factor into your research? Or are there people willing to help you out with this and say, hey, don't give me your data and we'll figure it out? I'm just curious what the state of the union is in your research world.
In terms of,
do you guys have models
using somebody
out there?
to
do you have an AI that helps you crunch specific proteomic data or mitochondrial data?
I don't do any of that. There are bioinformatics teams that do that work. I don't have the skills to do that work, to be honest. But yeah, I know I have the skills to say this is going to be an important technique. I can collect the data, do the data, get the data, hand it to somebody, and then I actually have to collaborate with somebody else that does that kind of work.
I gotcha. Is that sort of the standard operating procedure for how this works?
I mean, as scientists, we have, again, like I've said this term a few, but like we have our bag of tricks, and my bag of tricks is doing these aging studies, and and then that's why we have collaborators, so we don't have to reinvent the wheel. Like it would make sense for me to be like, well, I'm not an expert in epigenetics, so I'm going to just collaborate with someone who's an expert in epigenetics.
Yeah, for sure.
What's always funny to me is that every PhD I've spoken to, they are like the one group that's like, yeah, that's not me. You ask some dude on the street, he's got opinions about everything.
Yeah.
He's right.
Yeah.
And you're like, no, that's not me. That's not me. That's this guy over here. Let him have it.
So Emory funded your pilot. Do they then encourage you to sort of stick within the Emory family for like the epigeneticist and the data person and that, or do you guys have?
So I mean, Emory was instrumental in this process, and I started the studies. They funded it. I did the studies at Emory. In the process, like we finished the study there. Literally, the month we finished the study, I actually moved to Baylor College of Medicine, which is where I am now.
Nice.
So on the paper, because we did a lot of the analysis then at Baylor College of Medicine. So both universities get credit. Emory gets credit for the funding, but my affiliations at both go on the study. But yeah, I mean, it's up to me as a researcher to decide, who to collaborate. And really, what I like to do really is find whoever is the best person in that area. Who I like to work with. That's important. Finding cool collaborators. So it's been fun to have people reach out and say, oh, I can do this. Well, wow, I didn't even think about that idea. So the attention has actually helped the research progress in a way, because again, people are coming to me and saying, hey, what about this? And I can help you with that. And maybe we should put a project together on this. So that's been fun to see how just the media has kind of brought attention, which in turn have brought new collaborators to me.
That's great.
Yeah, and it seems like you've got more than enough homework on your desk to do 25 other studies out of the same data without running another nickel of actual lab work, right? I mean, you guys got.
I mean, it's funny because I didn't even think about it when I was doing it. That this could happen, but it was more like I just want to get this piece out because the other pieces are so complicated. I need like a lot more expertise to really understand and stand firm on what I'm gonna decide. These are the findings. Because a lot of as a scientist, it's about how we interpret the data, and again, I need help interpreting the data. But again, it's been incredible to have these new people come in from the attention and really help me interpret the data. And there's so much more to understand about what we have collected. So lots of exciting stuff coming down the pipeline.
That sounds awesome.
I absolutely love it. That is a perfect segue actually into, what are you up to now? Like, what do you got going on now? Walk us through what Fibernox is doing.
Asking. Yeah. I'm glad I have this opportunity to tell you a little bit about my company, Fibernox, and some of the work we're doing there. So as I mentioned earlier, my main expertise is actually in fibrosis, which again is the clinical term for scar tissue. And Fibernox, my company, I'm the CSO. We have a CEO named Joe Patty. And what we're doing is developing therapies for fibrotic disease.
Awesome.
And so my background in training, I have some weird kind of combination of graduate degrees. I have a master's degree in cell and developmental biology, a master's degree in ecology, evolution, and behavior, a PhD in applied physics. It sounds like, none of these things go together, but actually being this kind of jack of all trades scientist and being able to look at things from different angles has really helped me in terms of developing therapeutics.
I love when people say jack of all trades. They're like, I know plumbing.
I dabble
I know
with a PhD.
Yeah.
I was gonna say I'm sapiosexual, and you just went up like 5 more points in my book, and I don't want to be inappropriate because this will be the one my wife listens to, and she's like, You fucking hit on a doctor?
Yeah, like,
No. I was just saying.
Yeah. This is great. Yeah.
This is
That's
great.
quite the collection of accoutrement there. You're like, oh yeah, I've just got a couple of these masters and a PhD hanging around. Yeah.
You know, I just stick around over here.
Yeah.
Oh my goodness.
Hilarious. Yeah. Well, I mean, look, that you can kind of see how those are connected. So you were.
Yeah. So what the example I was going to say is, so evolution, I like to think about things from an evolutionary perspective. So fibrosis, scar tissue, is evolutionarily conserved, right?
Mhm.
If you were to fall and scrape your knee and you couldn't develop scar tissue, you wouldn't survive, because you need that. You wouldn't have the way to pull the wound closed, which is what these myofibroblast cells do. You wouldn't have that barrier to prevent infection. So we wouldn't be here without the ability to have scar tissue. So scar tissue, we need it. But at the same time, especially in the context of aging, scar tissue can become a pathological process. And so we have a lot of diseases of scar tissue in aging. Again, and it's a key component of end stage heart failures. A factoid I like to share about scar tissue that a lot of people don't recognize is that you're going to be shocked that almost half the deaths in this country are attributed to scar tissue.
Say what?
Yeah, exactly. That's the.
How is that?
What's
Is that the Red Hot Chili Peppers song, or?
Yeah.
Well, so that's the reason why people are not aware of this factoid is that the cause of death isn't scar tissue. You don't die. You die of organ failure. So like liver cirrhosis, and again, end stage you die of heart failure. But what is the contributing factor leading to that heart failure? In many cases, is scar tissue. So you have a you have a heart attack, and then where that myocardial infarction happens, that causes a scar, and that scar will grow forever. And so as your heart starts contracting more where the scar is, the other side of the heart enlarges and so your heart is constantly remodeling until ultimately you have heart failure. I study a rare disease called idiopathic pulmonary fibrosis, or IPF, which is one of the most fatal and progressive. It's the most progressive and fatal form of scar tissue. Average age of diagnosis 66. So most people only get it when they're older. And these patients just get this progressive scarring of the lung. And so there are 2 FDA approved drugs for this disease. And what they do is they slow the progression of lung decline. But they don't stop the disease. They don't reverse disease. They're not a cure. And so what we're doing at Fibernox that's, I think, really different and unique from other strategies, is we're targeting age-dependent pathological dysfunction. And I'm going to throw out a big word for you: antagonistic pleiotropy, which means that you can have something that is beneficial in early life, but becomes detrimental later in life. But because you're post-reproductive, it's not selected out of the population.
Oh, that's cool.
So NOX4 is a main target that we work on. So NOX4 is the master regulator of oxidant production. And one of my sort of claim to fame many years ago was the first to define that NOX4 dependent oxidant production is a key and core pathway for developing scar tissue. And so what happens in the context of aging is that in normal scarring and physiological scar tissue, NOX4 turns on, you generate ROS, and then as the injury is resolving, NOX4 turns back off. In aging, NOX4 turns on, generates all this ROS, reactive oxygen species, and then the switch that turns it off is broken.
Oh, wow.
And the same things happens with our other target. So we're developing NOX4 inhibitors to specifically target that age-dependent pathological dysfunction. And I personally think that those make the best drug targets, these antagonistic pleiotropic targets.
And the psilocybin stuff, is that sort of just help prevent getting there or extend that? Is that the tie-in there?
Right. So it's interesting. So I actually tested to see if psilocybin targets NOX4. And in our paper, we actually show that that long-term treatment in cells, it does reduce NOX4. But I tested, does it directly impact NOX4? No, it's a downstream effect of.
I see. Okay.
Oh, wow, cool.
So they're connected, but it doesn't. So we're developing inhibitors that directly target NOX4. And then, so we reduce oxidant production. And then on the flip side, we're developing NRF2 activators, which NRF2 is the master regulator of oxidant defenses. So if we can reduce oxidant production, and on the flip side, improve or enhance antioxidant responses, then we can directly target this massive oxidative stress problem that happens in the context of fibrosis.
You know, what's interesting about that is like you're targeting a specific disease process, but it could also have longevity implications.
I was just going to say, does Sinclair and those guys start using this stuff like in a completely different way, similar to like a rapamycin evolution or a metformin?
Yeah, it almost seems like
evolution?
rapamycin, where like the right dosage there is critical, because you know, if you get cut, you're screwed if you have too much.
Right. Yeah. I mean, oxidants are this thing where you need an optimal level. You don't have enough. I mean, we need oxidants for normal signaling, but too much is not good, right? So that's oxidative stress, and oxidative stress is really that balance between oxidant production and antioxidant responses. The problem with in aging, and especially in the context of disease, your oxidant production is higher, and your antioxidant responses are, at least for NRF2, which regulates this in scar tissue. Not only does it not work, but it turns off. It just goes the opposite, plummets. So
it's the worst of both situations.
Worst of both situations. Like dysfunctional antioxidant responses and boosted oxidant production. And so at Fibernox, we're developing NOX4 inhibitors and NRF2 activators to combat that oxidative stress issue. And we're also doing something, I think, unique that 10 years ago, people told me this was a terrible idea, and I ignored everybody, which was giving inhaled. I mean, the lung is one organ where you can deliver directly.
Highly vascular.
Yeah, exactly.
Yeah.
And
so, and actually we're starting to see now in the pipeline of clinical trials that there are a lot of therapies being developed that are inhaled for pulmonary fibrosis. So we can get it to the lungs at much higher.
Almost nebulize it, or are we talking like an inhaler? Yeah.
Yeah, Chris McDonald was on our show a while back, and he has this patented nebulizer technology, and I can't remember the exact diameter, but it's like whatever a regular, like, albuterol
nanoparticles.
Like, it was like nanoparticles, like really small. And basically, as a result, things like metagenine and like all the stuff like in kratom and all the THC stuff and all those things, they all basically got delivered instant to the bloodstream, like, because it basically is smaller than capillaries. It's just shooting right in there.
It's not always a drug show. It like a lot of times it ends up there.
Yeah. Well, I mean, Chris's whole thing is inhaling, right? So it's all about atomization, right?
But
like,
I love that too, because it's also a direct route. If you're trying to fix a lung disease, that's a pretty straight line.
It makes sense, right?
You're not overthinking that one.
You
put it in the lungs.
Right there.
But, at the time, people were thinking, when you have scarring of the lung, how
you're going to absorb it. Yeah.
Right. But we've shown that we can get it into the lung and actually reduce it, have a much reduced drug systemically, which, if you think about it, again,
that's great too.
Thinking about these evolutionarily conserved pathways, you really want to target where it's dysfunctional. And you'll have a lot of the problem with the current therapies is that 50% of patients discontinue treatment because of adverse effects. Pathways that we need for other things.
And are those mostly in the biologic realm? Are those the biologicals? Drugs, are they?
No, the current drugs are small molecule inhibitors.
Okay.
But they're targeting, sort of what I call initiation events in fibrosis. And we're targeting things that I think promote the resolution of fibrosis.
That's pretty
cool.
So
once you have it, you got to get past. Yeah, got it.
Yeah. And so one of the I think advantages of this strategy is that we can not only halt or at least reduce the progression of the disease, but we can actually, with these therapies, at least in mice, we can actually reduce the amount of fibrosis that's already there, which current therapies don't do.
Yeah, that's huge.
Yeah, and actually there's more than a couple fibrotic based bad diseases that you don't want to get. We have a family friend who just got diagnosed with myelofibrosis, I think it is. And like, I'd never even heard of this before, but we ended up doing some research into that. That it's my understanding that that's also fibrotic in nature, and it is not good. It's not a great.
Yeah, we don't have great therapies for fibrosis. I mean, we really just don't. And I think, again, part of that is that these are tough things to target because they are evolutionarily conserved. And that's why our approach at Fibernox is target that age dependent.
Yeah, that's cool.
That's very cool.
Yeah, that's awesome. So where could we send people to find out more about you and learn a little bit more about
and you also got to promise to
Fibernox?
come back as you keep uncovering cool shit.
To come back.
We'll find another. Maybe you can come on and talk to this guy we have in mind about no viruses exist. I mean, maybe.
You can find me. You can find out if you want to see more about Fibernox at fibernox.com. And you can see me on LinkedIn and on X. I try to post updates on my research there. Can't wait to post this on there.
I love that, in 2025, a research scientist doctor like yourself has got to be on X because you have to be. It's like
I still keep thinking people are telling me they're on ecstasy, and I'm like, okay, that's cool, but it's like middle of the day, man.
Yeah.
I don't really do social media personally. I don't have any, but you're right. I'm like, I had to get on that because you're behind in the times now if you're a scientist and you don't have those things. I'm trying to.
Yeah, it's almost like publisher Paris has changed a little. Huh?
Yeah,
well, it's also a good way to get your stuff out there. And hey, I just published this, and then you can share it with everybody, and it's actually a nice way to share that information.
And when you're working on the cool shit like the psilocybin thing, that thing is got instant virality built into it.
Well, I mean, that's kind of how I found you. You literally commented on one of our contacts, like, "Oh, that's my study," and I was like, "Holy shit, will you do our show?" And you were so gracious. So thank you.
Yeah.
Thank you so much.
Yeah, but it's just funny because 10 years ago, people would be like, what are you kidding me? I'm not getting a social media account to talk about my research. Are you nuts? Who does
It's published.
Yeah, it's published. Go to there.
In a real journal.
Yeah. Cool. Well, Louise, thank you so much again for coming. And we'll send everybody your way. Give us a minute and we'll put this out and hopefully we can tell everybody about you.
Awesome. Thanks so much for having me.
All right.
Thank
Take
you.
care, guys.
Be subversive, irreverent.
Health is wealth, but you knew that.
Everybody knows that.
But so is following us on social media at Irreverent Health. And check out our website for more exclusive content at irreverenthealth.com.
Oh, I like this.
Thanks for tuning into this episode of Irreverent Health.